right. Thank you everyone for joining today and welcome. For those of you who don't know me yet, I'm Alex Menz, the director of our Molecular and Genomics Informatics core. Today we have the pleasure of having the team from Seer on to talk about their proteomics platform, which will be a great seminar. But before I turn it over, just the general housekeeping items. If you have any questions, please put them into the chat or the QA box. We'll monitor it and answer anything we can live. Anything that we don't answer live, we'll answer at the end of the talk. And then we'll have a question session then as well. So with that, I'll turn it over to the Seer team and hear more about their platform.
All right, hi everybody. My name is Kevin Lawler. I'm the Senior Regional Business Manager for Seer. I cover New York City and New Jersey, so I'm your representative at Rutgers. We're very excited to share with you our Seer Proteograph product suite. Going through our presentation today is Mara. And I'll let her introduce herself.
Hi everyone. Sorry, I was just explaining earlier, my voice is a little raspy today. A little too much team spirit in our commercial meeting that we've got going on. But I'm Mara Riley. I'm a field application scientist based out of Alabama, and I work from Alabama all the way up to the New York, New Jersey area. So I'm also your local FAS. So today we're going to go through the Proteograph product suite and give you a little background on everything. But before I get started, I just wanted to give you even a little bit more info into kind of who I am and where I came from. So, oops, if I can PowerPoint and I get along today. So I actually, used to be in a lab, and that man in the middle in the white lab coat is Max Mahoney, who actually still works at Seer, and he was my FAS back in the day. So I was one of the, I think we were number 3 to get an instrument and work on it, and it was my favorite platform ever. I just loved what it could do, and kind of like where the company was going and what they were enabling for proteomics research, that when I had the chance, I just kind of like jumped at it to work for them. So I hope that you guys can, get as excited about it as I was as we go through this. So on our agenda today, I'm going to go, though most of you might have a proteomics background, we're going to touch on those current challenges in scaling proteomics for biomarker discovery. We'll give you an overview of our Proteograph product suite and everything that it entails, show you some of our exemplification data, and we will have time at the end for Q&A, but don't be, shy about asking questions as we go along. I'll be happy to stop and answer any that I can. And that's why I've got Kevin here to be like, Marsh, they want to ask a question. So we'll get on with that. But as we're going, I come across this quote a few years ago from Philip Geyer and Matthias Mann, that they said that among the first 300 highest abundant proteins, every 4th protein is a biomarker. But in the next 1,200 proteins, it's only every 25th protein. So this is really showing that we have so many biomarkers that still are to be found. And so we just really have like a long way to go. And so everyone understands the idea of the central dogma that you go from DNA to RNA to protein. We even have oh my goodness, sorry. To date, we have a really great understanding of the genomics of most living organisms, and that's kind of a great non-moving indication of health risk. Working at that DNA level is really easy now, because we have very well characterized tools to understand what's happening, because we've been sequencing now for over 50 years, starting all the way back with Sanger sequencing, to our tried and true favorites for most people using Illumina. And the problem is that when you move further down that chain, you get closer to that phenotype. And being close to the phenotype, there's been a huge push to try and understand more about transcriptomics. However, a lot of recent publications are currently showing that there isn't really a huge correlation between what's happening at the transcriptome level and what's happening at the protein level. So this kind of makes proteomics a little different and more you move up in complexity. So to truly understand what's happening at the phenotypic level, you need to get as close to the phenotype as possible. And which means, studying proteomics is absolutely necessary to being able to understand human health. So understanding human health, we can use a lot of different sample types, though most of the sample types can be incredibly invasive, like tissue biopsies. I've talked to a couple people that you get one tissue biopsy, you never want to have to go back for another one. But researchers can use them to analyze differentially regulated proteins and their association with human health and disease. But there's also non-invasive alternatives, including urine, saliva, and serum plasma, that allow early detection of disease and enable these long-scale longitudinal studies. So blood plasma, which includes signals from most tissues, is a rich source of proteins, biomarkers for disease detection, and it's one of the best options as your blood's traveling throughout your whole body. So keeping with plasma as our example, because that's kind of what Seer, though we're not a one-trick pony, that's kind of what we started with. So we have a lot of our data and a lot of our background is coming from plasma serum. Plasma has a large dynamic range of protein concentrations, which is presenting a huge challenge for any downstream detectors. So that large dynamic range, when you look at it, your top 22 proteins are making up 99% of your total protein mass. So that means a minority of the proteins are contributing to a majority of the signal. And the trade-off between that depth of coverage and the number of samples have made many discoveries impossible because of that. And particularly challenging are discoveries of the low abundant and multi-protein signatures that require the combination of depth and large-scale sample numbers. So in turn, if we can solve for that problem, hundreds of new biomarkers and new biology will be in reach for us. So down here, let's see if I can get a pointer. Okay, great. These dots show an accumulation of known biomarkers as a function of the depth of the proteome. The difference between these two lines here shows you the number of expected biomarker discoveries if we can access that depth in a scalable way. So as you can see, hundreds of new biomarkers could be discovered in plasma alone. And so that's really what we set out to solve at Seer. So we created the Proteograph product suite. It's our technology platform that we just broadly released January of 2022. And with our current iteration of our product, we enable you to see about 4 times more peptides and proteins in plasma with unbiased mass spec at scale. So let's get into a little bit more about how we do this. So at Seer, we introduce using nanoparticles. But what are these nanoparticles? Each nanoparticle is comprised of a super paramagnetic core, which makes it very easy to automate the entire sample handling process reproducibly. At a high level, we can tune these nanobio interactions at 2 different levels. So one, we can change the surface decoration of these particles with literally all known organic and inorganic chemistries. So I've done just a very small selection, as an example around this nanoparticle on the screen now, but it doesn't have to stop there. And then we can also change the corona formation conditions and competition between proteins for binding in the assay as well. So our technology is leveraging that interaction of the nanoparticles and the proteins that form, creating complexes that we call protein coronas. So in these coronas, proteins are captured based on their affinity, concentration, and competition for the nanoparticle. Importantly, this principle neither requires us to target a single protein epitopes, nor does it require us to know which proteins are actually in the sample. And another thing about our nanoparticles is that it's a common misconception that we're targeting or we're looking for specific proteins. That's not what it's actually doing, because we can use different nanoparticle corona formations. We can go after classes of nanoparticles, and that allows us to look at the albumins, but also the chemokines that are in the sample. So now that I've told you a little bit about what our nanoparticles are, what happens when you actually introduce them into the sample? So when a nanoparticle comes in contact with a biological sample like plasma, it samples proteins from across that entire dynamic range of the protein present in the sample. So when a subset of the proteins in the sample binds that nanoparticle surface, equilibrium is being approached. And as we're approaching that equilibrium, protein sampling is a function of 3 factors. So we're going with the concentration of the various proteins in the sample, affinity of those proteins for the nanoparticle surface, and also the affinity of those proteins for other proteins through protein-protein interactions. And the best thing is we don't have to stop at just 1 nanoparticle. As you can see here, adding 1 nanoparticle with a specific affinity gets you at certain parts through dynamic range, but when you add additional nanoparticles, we can keep sampling additional proteins across the entire dynamic range of the proteome without the need for complex workflows. So each particle is optimized for breadth while maintaining throughput. And this is all while performing independent sampling in a single well across the sample plate. So by using a panel of these chemically diverse nanoparticles, we can broadly interrogate across, again, the entire dynamic range of proteins and the biological sample with a high degree of precision. So now we can cover that whole dynamic range of the proteome, but that doesn't really solve the issue of when you think about the mass spec and being able to see all of that data. So I like to use this simplified graphic. Thank you. Gosh. So what do you do if you have a file on your computer that's too large to handle? Like, I know my boss is not going to, let me go and be like, I'm sorry, it's just too much work today, right? You could either not work with it, or you could just work on parts of the data. Again, nobody's just going to allow you to work on like a little part and then get away with it. So what we do, you can compress the information down and reduce the redundancy to something that's more manageable to work with. And that's what we do with our nanoparticles. So now a low abundant, high affinity chemokine can displace a fraction of the low affinity, high abundance albumin in the samples. And this may sound complex, and it is, but luckily we can draw from decades of experience. Nanomaterials have been engineered and explored in the context of nanomedicine for years. So it's really something that's been well studied, and it's very reproducible. So not only are we compressing the dynamic range to make it more usable, we are also compressing the workflow. So when we think about it, the top workflow here is the gold standard workflow for mass spec-based deep plasma proteomics, which includes abundant protein depletion and extensive peptide fractionation. Take weeks and require substantial analytical experience to perform them. So our nanoparticle-based solution, which replaces all of that upstream complex workflow, and it reduces hands-on time from days to hours. So you have that top workflow, and you're getting your thousands of proteins, and that's great. But who has weeks to sit and work? Because we're definitely in a society of where the more you can do, the better. And so nobody has weeks just to get the same data that you could from the Seer. It's broadly accessible. You're still getting thousands of proteins, but you're getting it in a work day. And that really, helps, it simplifies and automates the process, resulting into just like a whole solution. So we have our end-to-end solution, which offers consumables, which include the reagents, the labware, literally anything that you could need to run the instrument is contained inside of our assay kit. So next, we also have the instrumentation. And not only have we done scripts and automated it for our assay, we've also added things that enable us to do more with the automation, to where we can do peptide quantitation, get that assay set up ready for using your fluorimeter. We also, which is nice because anytime you can take pipetting out of my hands, like I know like I'm almost like carpal tunnel here from like all the pipetting I've done in my years. But it also can do reconstitution, but it's not that it's going to normalize to just one volume across the whole plate. It'll actually look at your quantitation data, and you can put your input max amount that you want to target for your mass spec, and it will normalize your plate at whatever volume that you need to make sure you're injecting at the correct amount for your mass spec. We are, like what I like to say, BYOMF. You can bring your favorite mass spec as long as it's high resolution. We're very agnostic, whether you know Thermo, Bruker, SciEx, anybody that you like to use. And then on the back end, we also offer a Proteograph analysis suite, which is if you're new to data analysis, or even if you've done it in a while, you know, it enables you to just get a clearer view of the proteome. And it's a great place to start with our graphics. And we'll talk more about the analysis suite in a little bit. So this is a very simplified version of how this whole process works. So we're going to still keep with that plasma reference per sample type going through. So you're going to have your plasma, and then it's going to be introduced to the nanoparticles. And we start with that particle matrix association, and it goes through an incubation where the sample and the nanoparticles are coming together. And during this incubation, because we've timed it out, it's giving, you know, time for those low abundant proteins that may have a higher affinity to the nanoparticles to come in and kind of elbow out and shoulder in, you know, to get the nanoparticle away from all those albumins. After that incubation is finished, we go on to the protein corona wash. So we're going to wash away anything that's not bound to the nanoparticles themselves. And after that washing, we're going to go on to our peptide preparation. So at this point, we do like a one-pot digestion. So we're doing the denaturation, the reduction, alkylation, and then digestion all in there. And so they'll incubate again for a set amount of time. And at the end of that incubation, we're going to do a mixed media filter plate cleanup to purify our sample. And then at the end of that, you're ready to go to the mass spec. And the great thing about all of this, again, is that it's automated. You only need to set up the instrument at the beginning of the day, and then you come back at the end of the day and you're ready to go. So I've told you about what we can do and how we can do it. But what does that look like when we compare to common proteomic workflows like I mentioned earlier? So to assess the performance of the Proteograph workflow compared to other commonly used workflows, we used a single control pooled plasma of 153 healthy and lung cancer patients with various ages, genders, and disease stages, and compared our 5 nanoparticle assay performance to neat digestion, depleted plasma, as well as traditional deep plasma proteomics workflow that included depletion and peptide fractionation together. So our data, as you can see, is demonstrating that Seer's Proteograph data is enabling significantly more protein groups, especially just compared to your neat digestion. And then when it comes to how deep we can go, I'm showing you here waterfall plots a waterfall plot where we are mapping identified proteins from each workflow against the human plasma proteome database. And these proteins are ranked in order of high abundance to low abundance from the left to right along the x-axis. So as you can see, we're really getting into that. Our 75th percentile is much further to the right, and we're really filling in that lower abundant area versus depletion and neat samples as well. Fractionation, you can get a little bit lower, but as you can see too, at the higher end, because you're depleting out those samples and those higher abundant proteins, you're kind of missing some data there. And it's great that we can do all things, but how precise is it actually? So as you can see, our CVs are right around depletion and much lower than fractionation. And of course, neat digestion is always going to be, a lower CV value, but that's because one, there's not as much sample manipulation going on. And also you're comparing your CVs, that precision, it has a much smaller group to look at when you're only getting 750 protein groups versus almost our 3,000 protein groups from plasma, from Seer's thing. So now I've given you data to show that we can see the proteins, but what does that do in a world of research, right? What biological insight can we actually gain from using the Proteograph? So non-small cell lung cancer is an excellent example of how translational and biomarker research can benefit from the utilization of this platform. So in using the Proteograph data. So worldwide, lung cancer is the second most commonly diagnosed cancer. About 80 to 85% of lung cancers are non-small cell lung cancer. And by the time it's diagnosed, the cancer has usually metastasized in 40% of its patients. And most patients, their diagnoses are at such an advanced age, they have a very poor prognosis. And the 5-year survival rate for people with non-small cell lung cancer is only 26%. So the use of a non-invasive sample like plasma for early detection can improve the identification, treatment, and prognosis of these non-small cell lung cancer patients. Sorry, one second. So here we did an unbiased proteomic profiling of 141 samples in our cohort from patients with non-small cell lung cancer, and we compared these to healthy and comorbid controls. So on average, we identified around 2,100 proteins per sample and 4,000 proteins across the whole cohort. And this whole study, from running the samples and the mass spec, only took us 2.5 weeks. So that's really fast comparatively to when you're thinking about how you traditionally do proteomics. And digging a little bit deeper into the data, on the left-hand side, we're arranging the data in an unsupervised hierarchical clustering map. We can clearly see that the differentially abundant proteins between the disease and healthy samples are clustering together. And then when we used data and machine learning, we were able to actually build a classifier with high specificity and sensitivity. And our AUC for this study was 0.96. And then one of my favorite biological insights that we gained from this is on the right-hand side. So we were looking at the data of the top protein biomarkers associated with identifying non-small cell lung cancer. 17 that we found were already shown in literature. So for us, that's a great indication that what we are doing is working and that we're seeing things that people have seen before. But at the same time, we also located 6 more that could be equally predictive or just as important as other protein biomarkers that have been found previously. And this obviously would have been extremely difficult to accomplish if we were using any approach that only targeted known biomarkers. So if you know what you're looking for, that's great. But if we're out here trying to, find better ways to, have earlier diagnosis, you really kind of just need to see what you don't know. So the other great thing is, I know I told you earlier that we weren't a one-trick pony and that we could do more than just plasma. So we started out with human plasma, and then we moved on to model organisms. So we did mouse serum, we've done pig. I mean, you've named it, we've probably tried it. But we've also moved on to other things, like including urine, CSF, conditioned media. There's things that are not shown on the slides, like we've done human milk. We've done homogenized muscles at this point. If you can put it and make it a biofluid, there is no reason that we have looked at to where it doesn't benefit from using our product. So kind of digging more into the second layer of this with our model organisms, again, this is showing you what we've studied to date. So we've done canine, porcine, bovine, non-human primates. But of course, most commonly used are mice. Now, the problem with mice is they're not very big. So with our workflow, you have to use 250 microliters because of the instrumentation. Now, with the mice, especially if you're doing a longitudinal aging study, you're not getting 250 microliters of mouse serum from a mouse without losing your whole mouse. So the great thing about that is that we've optimized a low volume protocol. So even if you have a low sample volume, we have a proprietary buffer in-house that you can add to your sample to dilute the sample to bring it up to that target volume so you're still able to use it. And looking at that here on the right-hand side when we're talking at protein groups is here's your 250 microliters and your 4x. Gosh, the people are in the way. And your 4x being better than the direct digest. But even doing 10 microliters, we're getting still almost 3.5 times more protein groups than your direct digestion. So that's enabling you to do these longitudinal mice studies if you want to. We're actually currently doing a 900 sample mouse study right now with longitudinal. They're using 20 microliters. So, I mean, this is definitely something that we have optimized and we're moving forward with, and our customers are also using as well. And they're getting great results. It's not just that we did it in-house and we're like, oh, look at our pretty data. They're getting the same data as we are. So it's very encouraging for us. And so moving on down to more sample types, we also have done neat conditioned media. So we analyzed the neat conditioned media in the presence of the bovine serum. So we didn't starve the cells. We gave them BSA and still processed them. And we processed them with the Proteograph and compared that result to neat conditioned media digest. And here you can see we analyzed it. And with our protein, we're still seeing 8.5 times more than just neat. And as you can see as well, especially with BSA, the depth of the Seer Proteograph assay is unrivaled compared to neat digestion. So really, again, we're not just plasma, we can do the CSF and the conditioned media and all of those different types. So again, we've gone through now the assay, what it includes. We talked about using the instrumentation. We got to skip over mass spec because you guys use what you like. But on the back end, we offer an intuitive data analysis package with various visualization tools, libraries, and other proteomic analysis from a QC perspective to a research perspective. So in offering pause, it is just a whole solution. And I'm only going to touch on a couple of things because honestly, pause in and of itself can be a whole nother seminar. And we just don't quite have the time to go into everything today, even though I would be excited to do it for you guys. So our pause includes automated data upload from your mass spec to your pause account. We have pre-configured data processing pipelines, using well-known search engines like Diane, Encyclopedia DIA, MS Fragger, Max Quant for DDA and DIA. It's easy to assess and manage your data storage, and our assay and LCMS performance assessment as well to do QC. But the best thing about this is, which I kind of skipped over, is that it's based in the cloud. So you don't have those long mass spec wait times doing your searches. And on the right, we can see where we just did 24 samples and 72 mass spec files. And in pause, searching that with library on your local instrument could take you about 7 hours versus 4 minutes with pause and library free. We went from days down to hours. And this is great because not only is it freeing you up time, if there's something, where something to go wrong in your script and you uploaded something wrong, right? You want to know in minutes that it didn't work versus waiting 8 days and then you just walk back up to your local machine and you're like, and that didn't work. And then where are you at, right? You have to wait another 8 days to see if it worked again. And I have a customer that we had 8,000 mass spec files and it took them only 2.5 days to analyze the data. So it's incredibly quick. And not only is it quick, just the biological insights that we can get through this are just amazing. So we talked about the non-small cell lung cancer before. So we took that data back from 2020 and we took our classifiers that we found, which took many experts, right? 25 experts, months to really dig into the data and see what we could find. So we put our blinders on when we released 9 clicks in 1 minute to be able to visualize the same data that we had found 2 years ago. And I am no bioinformatician and I regularly use pause, so I know the user interface is very friendly for even the most novice of us mass spec users. And that's another great thing because as Seer, we're just enabling everyone, from the mass spec savvy to the novice user that just want to dip their toes into the proteomic world, enabling you to be able to do the science and get these biological insights from it. So, okay. So again, like I just said, we're enabling any lab to take on large-scale proteomics, much the same as NGS products did for genomics. Our product is utilizing unique technology capabilities through an integrated, easy to implement and reproducible workflow. And it includes the consumables. Yeah.
So, I mean, one thing that was awesome, just to shout out to Hong Lee's group, as you mentioned, bring your own LCMS. That is awesome, great in hand with cores like Hong Li's group, the Center for Advanced Proteomics. He unfortunately couldn't make it today because he's at ABRF. But if you want to talk about any of the LCMS stuff, Hong is a great resource to talk about that. So a question about kind of sample types. I know you mentioned this is very agnostic, which is great to see. Have you worked with any kind of like environmental samples yet? Things like like wastewater, seawater, any other other fluid-like solutions that you can work with? Have you done anything like that, focusing on more bacterial proteins?
I don't think we have yet, but the great thing about Seer as well is we do have a POP, or our principal. Oh my gosh, what is it? Principal. Thank you. Proof of. This is why I bring Kevin. He keeps me straight. So it's our proof of principle program. So where if you're unsure whether this is going to work or not for you, we can always set you up, and we kind of do it two different ways. We can do it from start to finish, so we can take your samples, run them on our Proteographs internally, and do the mass spec for you, or we can send them back to you if you would like to do your own mass spec. And that way we can kind of, especially if it's, like if you just want to be like, oh, I want to see my plasma samples, we're like, let's save you the time. We have, years worth of plasma data right now. But if you're saying something like seawater or environmental samples that we haven't really researched a bunch, we'd be more than welcome to, more than happy to work with you to figure out, set up a experimental design and see what we can do for you.
Okay, so that'd be really cool. I know there's some researchers that are doing different environmental work, or even for like microbiomic research, take fecal pellets, solubilize them, and do something like that would be pretty cool. But yeah,
a general rule that we do have is as long as the biological matrix has their proteins in the native form and it's a complex matrix, there really is no reason it shouldn't work. Once you start having denatured proteins and things like that, the nanoparticle interaction does not quite work as efficiently. But for all intents and purposes, a complex biological mixture should work because almost every single one we've put on the Proteograph has worked, at different levels of success. But it's very rare that we find a biological mixture that just does not work.
Okay. Cool. Yeah, I mean, it'll be cool as we go past, metagenomics to metatranscriptomics to metaproteomics, which would be funky. Okay. Then another question about the kind of the analysis side of things, because that's where I really live. So, I've noticed things sometimes get a little bit funky beyond what is built into like a pre-scripted workflow. What kind of customization is available for PASS in terms of like, kind of passing it off to open source tools, or even building in custom like R or Python functions? Is there any kind of that customization available?
So inside we have like your normal, like, Diane, because we use Diane a lot. So, I mean, we have like our set, human Diane, but you can go in and customize, especially because you're more savvy, you can go in and customize and tweak here and there how you want it to read. But also there's a button, a function in there that you can export all of your data back out, and so that you can take it and go, elsewhere and do, your more in-depth analyses.
Okay, so it'd probably be great to do all of the kind of the labeling step through PASS and then pass it off to any kind of normalization you have to do or anything like that.
Correct. And then PASS is always, updating and doing new things as well, because like we're a new company, PASS is, newer company. PASS is also, it's not quite at its infancy stages, but, we're always looking for feedback from our consumers to what they like to see, so we can build it out and be, as user-friendly as it can be. Okay,
cool. That's great to see, and hopefully we'll see it continue to grow as Seer continues to grow.
Yeah.
Cool. That was all my questions. I don't see any in the chat yet. If anybody has any questions, last chance. Okay. I guess if nobody else has any questions, we'll end the seminar for today. But I just want to say thank you again to the Seer team for giving a great presentation. And we'll be sending this out over the next couple of days to anybody else who's interested. And if you have any questions, of course, contact me, Hong Li, or the Seer team directly, and we'll get you in touch with whoever you need to talk to about it. So thank you very much, everybody.
Thank you for having us. It was a great time to be able to present our platform to you guys.
All right. Have a great day.
Thank you so much.
Bye.